which is why she just said whatever she said on your computer. Okay, so I'd like to welcome everybody to our third week of office hours with Dave and Anita, and we have two guests this week that I'd like to introduce. Our first guest is Professor Dario Cantu in the Department of Viticulture and Enology at UC Davis. Dario is a native of Milan in Italy. He got his bachelor's and master's degrees at the University of Milan in agricultural science. And after that, he came to UC Davis, where he got his PhD in plant biology, working on tomatoes and disease. Following that, he joined George Dukowski's lab in plant sciences here at UC Davis, where he worked on wheat and diseases of wheat, and then joined our department in 2012, so about 7.5 years ago. In those 7.5 years, he went from never having worked on grapes before to being, I think, one of, if not the world's expert on grape genetics and genomics. He works on grapes and diseases of grapes, and is now working on more in the area, also in the area of grape flavor. We also have Dario's PhD student, Jerry Lin. Jerry got his bachelor's in plant biology and molecular toxicology at UC Berkeley, the other UC, and came here to get his PhD in horticulture and agronomy. Has been working with Dario for a few years now. This is really a very interesting topic because we asked Dario and Jerry to come and talk about a special project they have looking at a series of crosses between Cabernet and Riesling, which sounds like a weird cross to someone like me who's a chemical engineer. But the real power of this is not in the grape varieties that come out, but in the genetic knowledge. And this cross is going to put us in a much better position to understand the genetics of flavor and flavor production in grapes. And that's why it's so important, and why we wanted Dario and Jerry to actually talk about their project and answer questions. So with that, I'm going to turn it over to Dario to give a little bit of an introduction. I think Jerry has some information he'll talk about too, and then we'll open it up to questions as always. So Dario, turn it over to you.
Thank you, Dave. I will give the stage in a few seconds to Jerry. So Jerry will take care of all the background information on the project. I would just like to spend a few seconds introducing Jerry. Jerry has been in my lab for 4 years now. So he took his master's in viticulture enology first, and he was working on ESCA. And since a couple of years ago, he started this project on the genetics of aroma. And I don't like to contradict my chair, but we can also discuss about the potential of this project in developing new cultivars. So it's not only knowledge here. We can discuss also about how we can use what we're growing in the field. And Jerry, can you go ahead and share your slides so we can start?
Yes.
I don't know if Anita wants to say something.
Dario, you are welcome to contradict me whenever you want. You're the expert.
I got tenure now, so I can
You can do whatever you want.
All right. Should I begin or?
Please do.
Okay. All right. So I'm here to talk a little bit about my project. I've titled it An Integrated Genetic Approach to Wine Aroma. And sorry, this isn't letting me skip. There we go. So it's just a brief outline. I'll give a little background, explain the experimental design and the main objectives. So what we perceive of as aroma is the brain's interpretation of signals that are produced when volatile compounds bind to receptors in our nose. And these volatile compounds in the context of wine can come from the grapes themselves, the fermentation microbes, the aging, storage conditions. But for this talk, I'm going to focus on those coming from grapes. And the major compound classes that are important in wine and grape aroma are shown in the table on the left, with a representative compound, a common descriptor, and a cultivar where they are important. And as you can see from here, we know quite a bit about the compounds that are involved in aroma, as well as their precursors in the grape. And we know some about their biosynthesis, but we know very little about the genetics involved. And so there are a number of factors that affect grape and wine aroma, primarily the variety or cultivar, which you can interpret as a genetic component. The climate, viticultural practices, pests and diseases, those constitute the environment. And then fermentation microbes and winemaking practices either add new compounds or they modify ones already existing. And of these factors, the one I think we understand the least about is the genetics. And so that's my main research question. How do genetic factors contribute to cultivar differences in aroma? And to answer that, we're working with a Riesling by Cabernet Sauvignon population. This was a result of a cross done in 1994 by Dr. Andy Walker in our department. And the vines were planted out in the Wolf Scale experimental orchard in Winters. There's 138 progeny still surviving. And these two parents were chosen, I think, mostly because they are both very important cultivars for the wine industry. And they are very different in terms of morphology, how they grow, and their metabolites. And you can see that the table below, Riesling has moderately high levels of monoterpenes. Those are compounds that contribute like a floral aroma to wine. These compounds are almost completely absent in Cabernet Sauvignon. And it's the opposite story with methoxypyrazines, which are the green bell pepper aromas. And because the original population was planted just a single vine of each progeny in Winters, and they're not being maintained for fruit production. We were very fortunate to be able to set up a replicated experimental vineyard at the Oakville Research Station, where we have 9 replicates of each progeny in the rectangle shown there. That's the location of the vineyard. And so I began grafting these vines in the winter of 2017, rooted them and planted them out in nursery rows that spring, bare rooted the next year, and we planted those in May of 2018. And this is what they look like now. This is their third year, or their third season in the ground. And we are looking forward to having the first crop this year. And so the main objective of this project is to identify the genetic bases of phenotypic traits, most importantly for me, the aroma, but also other important traits. And I'll do that by first documenting the variation that exists in this population, most of which hasn't really been looked at prior to this experiment. And then associate genetic regions with traits of interest, and ultimately identify specific genes, alleles, or expression patterns related to trait variation. And so I'll show a little bit of preliminary data. So this is, like I said, the first year we'll be getting fruit. So some of the fruit data from here is from the original population. But already with the phenological data, so the flowering and bud break, we can already see a wide variation in the population. These 3 pictures here were taken on the same day last week, and you can see that some of the genotypes are already in full bloom, some are just starting, and some have a ways to go before they flower. Likewise, this is a bud break data that we collected this spring from the Oakville Vineyard. So this was over roughly a month in time, and you can see that the Riesling breaks dormancy quite early on the left side of the graph, and Cabernet is much later, and most of the progeny falls somewhere in between the two parents. And using this quantitative trait data, we can conduct an association between the phenotype, so the date, the degree days until bud break, with the genome. And I was able to find a marker on chromosome 4, shown by the red arrow on the right, that is significantly associated with bud break time. And so this is the type of analysis I'll be doing with the phenotype data I collect. And so other than phenological variation, there's also a lot of morphological variation in this population. And so there's more differences in the leaves. Some genotypes have leaves that look like the Riesling parent, others leaves that look more like the Cab parent, and some that don't look like either. And we can see the same thing in the fruit as well. With regards to cluster architecture, there are differences in the number of wings, differences in the size of the clusters between genotypes, as well as the density and of the berries. And among the berries, we have differences in berry size, and also with regards to seed number. And as for metabolites, that's what I'm most interested in. And we, other than the fact that there's color, that presence and absence of color in some genotypes, we don't really know much about the metabolic variation. And for the metabolite phenotyping, in collaboration with the Ebler lab, we'll be looking at the volatile profiles in the berries. And because both parents are wine cultivars and the winemaking process does affect the profile of the compound profile, we'll be making wine from these plants and analyzing the volatile profile there. And because the concentration of a compound doesn't necessarily correspond exactly with sensory perception, we'll also be conducting a descriptive analysis of these wines with the Heyman lab. And so that's just a brief overview of the whole project. If you guys have any questions, yeah.
Great.
So I'm going to start with a couple of questions, but I just want to mention for somebody that haven't joined us before for office hours, that if you press on participants, you'll see there you can raise your hand physically, or you can click a button at the bottom that says raise your hand, or in the chat box, you can write any questions that you may have, and we will repeat those questions to either Dario or Jerry so that they can answer them. Okay, so.
So Anita, before you start, maybe Jerry, if you can unshare your screen and that way people can see you better. That'd be great.
That's also a good idea. So I'm going to direct my first question to Dario. So Dario, you touched a little bit upon this in the introduction, but what's the overall goal of this project? And basically, what do you think would be the benefits for, you know, both us researchers that's interested in basic knowledge, as well as the industry as a whole?
Okay, thank you, Anita. That's a good point. So let's put into context why we're doing this. I think Jerry made a very nice case that we know a lot about the metabolites that are in the fruit and how they impact the aroma and flavor profiles of the wines. However, we know very, very little about the genetics, the genes that are responsible for those traits. So there is obviously a need of increasing the knowledge, filling the knowledge gaps that exist in the literature. And that's what wakes me up in the morning is learning more about biological system, and that's why I'm doing it. However, the more we understand the genetics, the more we can improve viticulture, in the sense that the more we understand what genes are responsible for aroma compounds, the more we can monitor those genes in the vineyard, and the more we can look and understand how viticulture practices impact the activities of those genes. So there is a clear, many of you are familiar with the literature of, canopy management, shading techniques, etc. Most of them look at the expression of genes at the end of the day. They look at genes in the phenylpropanoid pathway and how flavonoids, anthocyanin accumulate by looking at gene expression. So the goal is to be able to expand our capabilities of looking at fine detail how berries are developing as a consequence of whatever treatment we are imposing in the vineyard, with all the tools that we can develop with these projects and others. So there is increase of understanding, building new tools. And then I would say that, again, Jerry made a good case with all the picture that he showed of the different clusters. I think it's really impressive how just a single cross can unlock and combine genetic diversity that was unseen before the cross was made. We all complain about Riesling being too compact in its clusters and very susceptible to molds. And one cross with Cabernet, we can get very nice Riesling-looking berries with a cluster that looks pretty much like Cabernet Sauvignon, which we all appreciate because they're typically large in size and loose in density. So I think this is a clear example where we are, we are part of an industry where we don't take advantage of genetic diversity. We don't take advantage of genetic diversity in terms of exploration of the standing germplasm that exists all over the world, because we focus on few varieties. But also we limit ourselves because we don't actually enjoy the power of breeding and genetics that other crops actually enjoy on a regular basis. Thinking about field crops, you change variety every couple of years because you always grow the more adapted and the better one for the grower and the better one for the consumers as well. So I think my goal with all this is to be able in a couple of years to make wine tastings of all these lines in Napa, and with the idea to showcase the power of genetic blending, I like to call it, and the diversity of bud break window that we have in the population is also fantastic. The window of flowering time. Eventually we could have a mix of flavor, but also a better choice of phenology and of the variety we cultivate, depending where we cultivate them. So that's, I guess, where we're going with this.
Something I just looked at the varietal characteristics, aroma characteristics, and the first thing is that Riesling doesn't really have methoxypyrazines, where Cab have plenty. And I thought, wouldn't that be neat to have a Cabernet Sauvignon-like grape without methoxypyrazines?
Yeah, okay, we could have also a Riesling with TDN and methoxypyrazines.
Well, that would be the bad one, according to me.
Of course, but that's, the thing to keep in mind always that each variety is diploid and heterozygous, is derived from a cross. Mm. So, for example, Cabernet Sauvignon is a Cabernet Franc by Sauvignon Blanc cross itself. So yes. When you cross Cabernet Sauvignon, you are not only crossing Cabernet Sauvignon, you're crossing the two parents of Cabernet Sauvignon. So you expose a certain degree of genetic diversity that was hidden before. Because
yes, so you're harvesting the grandchildren.
Yes, in a way we're harvesting grandchildren of a quite, as they mentioned, a quite special cross, because Riesling and Cab are very distinct, and I think that was the vision of Andy Walker. Really try to maximize the degree of genetic diversity we can create by making a cross. You maximize this by picking very distinct parents. If you cross Cabernet Sauvignon and Cabernet Franc, you will see something there, but will not be as diverse as we are experiencing with this particular cross. Yeah.
So, Dario, follow-up question. So how does this integrate with the current breeding programs at UC Davis? And I was also wondering what's happening around the world regarding crosses and things like this.
So, as you know, my interest is mostly on disease, and I'm part of collaborations with Andy Walker, and you know very well Andy Walker's program. Most of the breeding going on at UC Davis right now is for disease resistance, which makes a lot of sense. both for the rootstock and the scion, being this mostly powdery mildew and pierce disease for the scion and soil-borne pest for the rootstock. And I would say that 90% of the breeding programs around the world have the same focus, particularly now in Europe, where there are restrictions to fungicide applications, and really sustainable viticulture depends on the development of varieties that are better adapted to disease and other stresses. However, when I think about disease, in a way is a yes or no type of question. You are resistant or not, and that yes or no resistance depend most of the time on introducing in the parentage of a variety another species. So we do these interspecific crosses, introgress into the variety resistance, and then you have acquired tolerance to the disease. One risk with just breeding for resistance is that you forget about all the other important traits of your wine grapes. And I don't know if you're familiar with the strawberries in California. I mean, strawberries in California don't taste very well and haven't tasted very well for a long time because the major focus was disease and shelf life. And we all know as winemakers, we cannot allow that. It would be very nice not to spray sulfur or other fungicide anymore. But if the berries are not good for winemaking purpose, then what's the purpose? So the idea here is to be able to generate information that can go together with all the breeding that has been done for disease to select, as we select resistant varieties, also select the better ones for flavor. And the beauty of doing genetics assisted breeding is that we can select varieties at the seedling stage. We don't have to grow them for 5 years to get the wines out of it. So the idea with Jerry's work, we will be able to have those genetic markers that we can use to predict the aroma profiles of the grapes before we see the grapes. And so advance the genetic improvement of new varieties, incorporating all the possible traits that we think are valuable. I mean, nowadays at UC Davis and other places, major focus is tolerance to drought, tolerance to heat waves. And so we cannot just think in compartments. Aroma has to go with disease resistance, with resistance to biotic stresses and so forth. I don't know if I...
Very true. Very, very good point. So Jerry, before you fall asleep, I'm going to ask you a question. So you mentioned about, all the morphological and phenological measurements you're taking, and that you're collaborating with the Evo lab to do analysis on the metabolites, and you're also collaborating with the Heyman lab to do the sensory analysis. And there's a lot of things showing some correlation between wine aromatic profiles and sensory. How are you going to connect the grape composition with the wine aroma? That's another step.
So part of the what I'll be measuring in the grapes is not just the volatile compounds, but also the non-volatile precursors in the grape. And for many of the compounds that are important for wine sensory attributes, we know what the non-volatile precursors are. And so that's, so I'll be trying to measure those as best as we can in the berry. And so yeah, though, to better have an idea of like how the grape and wine profiles will correlate.
I'm assuming you're going to make them all under identical winemaking conditions, because we know, like, depending on yeast and other conditions, yeast precursors, the grape precursors can actually have a different impact depending on the yeast sometimes.
Mm-hmm. Yeah, so that's an interesting issue we've been thinking about, is half the progeny are white and half the progeny are red, and how to best address that, given that it's like in an ideal situation, we would make 2 wines from each genotype, like a skin contact red or white, and then just a white or rosé. But I think if I'm gonna pick a metabolite class to focus on, maybe the aromatics, most of those are found in the skin, so it makes more sense to make all the progeny as sort of a red wine or an orange wine, I guess, for the whites. But another complicating factor is we've noticed that some of the reds are teinturier, so they also have some anthocyanin in the juice besides the skin. And yeah, that's gonna be interesting.
Gianni, you have a question? Just unmute yourself. Gianni, you're muted. Aha, there you go.
I'm gonna try. Listen, along the same lines of getting our heads or arms around all the variables associated with not only the growing of the plants and the fruit, but also the winemaking and the sensory side. When you look at I guess I'm trying to step back and see how large this mountain is. So what sort of infrastructure do we need to have for us to be able to not only collect all this data, but structure in a way that it actually is going to make sense later on, and then start deducing some pretty powerful things that we normally do today. So could you, Dario, could you or Jerry talk about what infrastructure do you either have in place or you're trying to put in place so that, you know, we start bootstrapping ourselves into this huge mountain that we're going to be confronted as we go with time?
Yeah, that's a good point. And I think one thing that Jerry didn't mention is the size of the population. So we're dealing with about 140 progeny, which are triplicated in 3 randomized blocks. So we have nine plants per genotype. So you can make the calculation. It's a big experiment. And for some observations, we're gonna take observations per plant, like the data for bud break and flowering or the phenology. The chemistry will be done per plant with the correct application. We cannot. There is no way we can do 1,000 wines and taste them all. I mean, we could do the wines, I guess, if we can scale up, but we will not ever convince Professor Heyman to help us to do a wine tasting with more than 15 or 20 wines. So that's, this is a project that goes in steps. So the first step for us is really appreciate the diversity from a viticulture standpoint, and that's what Jerry focus has been this year. Next will be to understand the variability in the chemistry of the juice. And this is something we're going to be doing with Professor Ibler. In particular, we're going to be looking at terpenes. So we're not going to look at all the chemistry of the berry. We're going to focus on specific compounds, which are important, particularly for the aroma inherited from the Riesling parent. So as you can see, we need to focus. Obviously, this is when I convinced the faculty to support this project, and that's something we should acknowledge, the fact that this project would not be possible if the faculty did not support investing part of the Oakville Station to this particular experiment. But the idea here is that we developed a playground where Jerry can play and understand terpene metabolism and make some wines out of it and understand the impact on the sensory responses to those metabolite concentrations. But it's, the population is a playground for much more. There are questions we could ask if we had the funding about microbial associations, how the genetics of the vine impact the yeast, the native yeast that are on the berries, and therefore how they will impact native fermentations. We can look at water use efficiency, and I know the new faculty in the department are already looking into that. How in this population water is being efficiently or not used by each individual, and so forth. So you're right, Johnny. It's a big mountain, but we don't want to climb the whole mountain in 1 year. We're gonna climb a piece of it, and so maybe something worth mentioning is Jerry and I last year wrote a review article that was published in Horticulture Research. The title is "The Genetic Basis of Wine and Grape Aroma," where Jerry did a fantastic job because he basically looked at the literature of the past 10, 20 years and identify critical gaps in knowledge of genetics of aroma. And those gaps in knowledge, and some of them are related to terpenes, are the focus of our research. And I think that's a good resource to have, but also to understand where we're going with this work. I don't know, Johnny, if I answer your question, if you have.
No, I really appreciate your thoughts, and this reminds me of a long time ago when I started in the processing industry, and we did not have the PC, by the way. We had the mainframes, and we wanted to take a process from raw materials all the way to the bumpers that we're making on our cars. And I was faced with a situation of saying, okay, here's where I am today. I don't have a PC, but eventually I need to have the capability for looking at the raw materials, the monomers, the polymers, the compounding, and then the bumper at the end. So I guess what I'm saying is, I think it's a good conversation to engage in terms of really how do we get our arms in this. We're talking about mountains. I think we're talking about oceans of data here now.
Okay, so to that, and I understand your question better. I'm proud to say that my lab is quite experienced with big data. You didn't have a PC. I have 4 servers dedicated to my lab, and we own part of the HPC of the college. So I think we're well placed in that sense, at least from a computational analytical standpoint. Our collaborators are fantastic. I don't know, Hibler has all the capabilities to process thousands of samples, and most importantly, she's willing to do that with us. And then I would go back to my answer before, and then we should be reasonable. and I think that's something very important. When you start projects like this, you should go to the library and look what are the gaps in knowledge and what your goal should be. And obviously, I like to think that I also work for the industry, the grape industry, so try to tackle something that can have a clear impact in the short and long term on the industry.
So Dario and Jerry, we have a couple more questions that are in the chat. Maybe I can read them. And I will point out that not only is Dario doing a great job with big data, but Mason Earls, who you hear from next week, is also working a lot on big data. I've done that, and some of our other faculty have as well. And it's been increasingly critical, if not essential, to have those kind of capabilities. So it's really important. So question was, why focus on terpenes over pyrazines first?
Maybe Jerry can follow up, but my take, we know a lot about methoxypyrazine synthesis. The gene or the gene family involved in methoxypyrazine, at least the final step, is known. So there are good markers to use to select varieties with or without methoxypyrazine. And so the terpene is really the area where we know very, very little. Jerry, I don't know if you wanna...
Yeah, so terpenes have been pretty well studied in muscats. We know the specific mutation that causes higher terpene production in muscats, but we don't know what mutation Riesling has, or we don't know why Riesling produces more terpenes than most other grapes, but less than muscats. And Also, the inheritance of the terpene trait from Riesling is also unknown. Methoxypyrazines, it's a dominant, I believe, trait. So all the progeny are likely to have some amount of methoxypyrazines from Cab. And but with the Riesling, the terpene levels are pretty much unknown. And also Riesling progeny, a few of them have been adopted or have become commercial cultivars in a few countries. And it'll be interesting to be able to see how these crosses compare with those.
So, but I think one of the things I would point out, and maybe you guys can, one or both of you can comment on this, is that, and I know this from our work with yeast, while the end pathways may be known, if you're changing a large amount of the genome of a grape variety, you could be redistributing fluxes through different metabolic pathways that would have consequences in flavor as well, even if that end pathway isn't changed, right?
Yeah, the tricky part with aroma compounds that are very potent at very low concentration. So we're looking at really nanomolar, for some even lower concentration. So it's trickier, than primary or even secondary metabolism, more in the central pathways. And so that there is a lot, some of them are produced but are not volatile. They're bound to sugars and they're not in the aroma spectrum. Yeah, it's not simple.
So this is a question for Dario from one of the participants. Since introducing new wine grape varieties is not easy, do you think you can use the information obtained from this research in, number one, editing existing varieties, or number two, screening clones within current varieties? Also, will the information from this cross be useful for other varieties as well?
Yeah, I mean, I like the idea of editing. Nowadays, it's easier to edit when you want to knock out genes. There are ways you could introduce genes or augment the expression of genes, but it's trickier. So editing is right now for grapes is more like a research tool than really a breeding tool. However, I mean, the more we understand the pathways, the more likely that at one point we'll be able to modify them with this tool. Concerning, of course, I mean, the genes that we identify in Riesling or Cabernet Sauvignon can help explore the genetic diversity across the germplasm. So if we have if we identify a gene, for example, gene X, that is responsible for terpene synthesis in Riesling, we can take that gene information and screen the entire germplasm to identify, without having to taste the wine, identify varieties that have the same form of the gene, of different forms of that particular gene, and prioritize those varieties for further analysis. Yeah, so knowledge is important, and once we have it, then it can be used in many different ways.
Good.
There was a second part of the question, or?
So I think, well, let's see. Could you use it to screen clones from current varieties?
Yes, yes. I mean, that's the answer would be the same I gave for other varieties. The degree of variability you identify in clonal selections is much smaller than what you have among varieties. So yeah, it's possible. Yes.
So this is another question. This is from Rodrigo, and he says, "Hi, Dr. Cantu. I'm from Argentina. I wonder if in the hundreds of populations of Malbec in Argentina, is it possible to find genes that control for disease resistance as opposed to varieties where diversity has been lost? Or is this unlikely?"
That's a good question. I would say never lose hope. Again, the broader the genetic diversity, the more likely is that you identify individual or subpopulation that have evolved to resist to a pathogen. So that's why oftentimes when we look for sources of this genetic resistance, we have to go outside of the Vitis vinifera species. Most of the genes that have been used to breed for genetic resistance come from other species. Those are wild species from North America, Central Asia. It's very hard to identify strong sources of resistance in close relatives of vinifera. You have observations in the literature of clones that are more or less tolerant to powdery mildew, for example, or trunk diseases, etc. So it's possible. It would not be the dramatic increase in resistance that you would see when you look at other species, but certain degree of tolerance. Sometimes you have a mutation, your leaves become hairy in a variety that was not hairy, and now all of a sudden, this particular clone is, the powdery mildew has harder time to attach to those leaves. So it will call that clone more tolerant to powdery mildew, but it's just the disease has harder time to colonize. So subtle changes can have impact on disease. So,
As kind of a follow-up to that, can you comment on, are there varieties out there that have way more natural variation than others, at least in terms of clones or even within a clone? Is Malbec more, the clones of Malbec more variable than, let's say, a Chardonnay or Cabernet in California?
Okay, so my answer, I don't want to sound bitter, but I think there is a need of more studies of clonal variability than we have in the literature. So oftentimes, we think clones are very diverse, but then you grow them in the same vineyard, and they don't display the same degree of diversity that they would if they're grown in different environments. So there's a very strong environmental component. We think there is also a very strong epigenetic component, but I will not go into that. Said that, there are some observations that Zinfandel clones are less variable than other, for example. Pinot Noir, Pinot Blanc, Pinot Gris are clonal variant of Pinot Noir, which suggests that Pinot is somehow more variable when you look at clonal populations, at least for the movement of the genes that are responsible for the coloration of the skin. So there are some observation that certain varieties are more variable genetically than others, but definitely we need more studies that look at the clonal population in the same environment, so determine what is actually the genetic component of that variability. And more study to understand why, if there is a genetic component or epigenetic component in the differences in variability between varieties.
So certainly, Rodrigo, feel free to contact Dario and discuss that idea more if you have a lot of diverse populations of Malbec.
Yeah, please.
So that leads me to my next question is, and either you or Jerry, Dario, so what do you think is the environmental contribution to the traits that you're measuring in your current populations? And are you planning on testing it under different environments, or are you trying to control the environment under which you're testing it?
Yeah, so we have designed experiments so the environmental component is taken off from the picture. That's why we had 3 randomized blocks with replicated vines. The environmental component is important and is always part of any genetic study. But if you really want to understand the genetics, you need to get rid of the environment. If you want to look at environmental component, then it gets complicated, because whereas the genetic component is one, the environmental component, which environment we prioritize? We planted in Oakville thinking that Cabernet Sauvignon genes will have their best chances of express their functions. Is not obviously the best for Riesling. So if I had to grow this population in another region, I would pick a region where Riesling genes will have the better environment express themselves. But that gets complicated because, then what is the third environment, the fourth environment, the fifth environment? We're talking about 1,200 individual population that need to be planted, grown in comparable ways, managed in comparable ways across environment. So for me, going back to Gianni's mountain, it gets really not even a mountain. Is trying to go to Mars. So for in that sense, let's start from the genetics. Once you get to the point that you know the genes of a pathway, then you can become more ambitious. You can decide, okay, let's look at those genes in different environments, just those genes. What is the expression under different temperature, different day lengths, etc. And that's possible, but I would see that as a follow-up study and not as part of this first observation. Yeah.
So I have and again, if you have questions, feel free to raise your hand electronically or whatever, or put something in chat. We'll read your question. But I do have a question, I think, for Jerry. So you probably, I know that your vines are just bearing fruit for the first time this year, but you've obviously looked at the fruit up until this point, or from the other vineyard. Mm-hmm. Do you have a sense for how much variation there is in time to ripeness, and how are you going to deal with that as you go to start to make wines from these grapes?
Yeah, so last year I looked at the ripeness, or the ripening dates for the winter's population, and so those plants aren't replicated, and also not managed in a typical vineyard setting, so it's hard to extrapolate. But from that data, it looks like we're seeing about like a 6-week window for each genotype reaching 22 bricks. So I just recording the date when each one reached 22 bricks, and so it's a pretty normal distribution across those 6 weeks. And if that holds true in Oakville, that would be great for sampling purposes for fruit and for winemaking, because I'll have maybe 10 genotypes ripening a week or a day, instead of having to harvest all 140 genotypes at the same time.
How much fruit do you expect to get from the 9 vines, just out of curiosity?
That is also very variable between genotypes. There are some genotypes where the cluster is about like 5 centimeters long, and there are some which I think I weighed, they were about 1 kilo each. So it's going to be a very big range.
Will you have enough fruit to make wine in our small TJ fermenters, or will it be a smaller scale than that?
I think smaller scale. Talking with Professor Heyman, we're thinking we need about 10 gallons to do a sensory study, and so that would be about the volume we're going to look for. And I think some genotypes, because we can't make all 1,000 wines, some genotypes will just be ruled off right away just because they don't produce enough fruit. And I know I have a few genotypes which haven't ever seemed to make fruit. So, yeah.
So, Jerry, you mentioned in the beginning Riesling and Cabernet Sauvignon, because they're both popular varieties in California. But, Riesling and Napa, not so much. I've Riesling myself. So any other specific reason why Riesling and Cabernet Sauvignon?
So I think part of it is because Riesling is very distinctive aromatically. It's one of a very few number of cultivars that produces a lot of TDN, and the cause of that is still pretty unknown. And yeah, and also Riesling, so I think Riesling shares Gouais Blanc as a parent with tens of other varieties. And so having that shared genetic background makes, like if we identify genetic variants in this population, we're likely to see shared variants in other important varieties as well.
And what's the other parent?
The other parent is, I think, so far unknown, but possibly related to Traminer, and which also is in the background of probably the majority of commercially important varieties in France. So
we're going to take a brief break here just to actually do an advertisement for next week's office hours. And so next week, we're very lucky to have our newest faculty member joining us. That's Mason Earls, along with Con Curterall, who's our cooperative extension specialist in viticulture based out at Oakville. So Mason is an expert in machine learning and artificial intelligence, in addition to plant physiology. He joined us in September. He's working on robotics as well. So we have autonomous robots wandering around our vineyards already. Joint project between him and colleagues in bio and ag engineering as well. And Con has worked quite a bit on vineyard mechanization automation as well, both in Central Valley and the North Coast. And they're going to be coming on together next week to talk about machine learning, artificial intelligence, robotics, vineyard mechanization, automation, all these things that are tools that will help us to not only grow better grapes and make better wine, but also to address the real labor issues that we're having in California and other places where we're growing grapes. So that's just a brief advertisement for next week. Does anybody else have any other questions that they'd like to ask our guests?
If not, I would like to mention that we did, like Dave said, we did record this session, and eventually we will have this loaded onto some site, and we will send the link to the different webinars so that you can access it remotely, or tell somebody else that missed this time zone but would like to see the discussion.
I do have a question for Jerry. So because you're probably the person who's spent the most time with these crosses, and I can't imagine that you wouldn't try tasting them. Have you found ones that have really unique flavors they haven't tasted in grapes yet, or ones that you think are going to make terrific wine? What's your thoughts on that?
I haven't tasted any that would make, well, that I'm not that experienced in this regard, that would make a terrific wine, but I've definitely tasted some very interesting ones, some with very high methoxypyrazine levels. So even when they are like completely ripe, they still taste like green bell peppers. And yeah, I mean, if they also inherited the terpenes from Riesling, I think that would make a very strange combination of flavors.
Any red grapes without pyrazine that have a lot of the terpenes in them?
I don't know, because I think this is true for Riesling as well, but most of the terpenes are in bound form, so you can't really detect them just from the fruit itself. It's when it's made into wine you can get that flavor.
Yes, just because the berries doesn't taste that well doesn't always predict that the wine isn't going to be that great either. That's good news. It's a pretty unpredictable.
Yeah.
Okay, so anybody else that thinks of questions later can either email Jerry or Dario directly, or they can email myself, David Block, or Karen Block, and we will make sure that the questions gets directed to the correct person, so that your questions may get answered. But otherwise, I'm gonna throw the ball to the chair.
Yeah, so I want to thank Dario and Jerry for joining us today and answering questions. I want to thank Karen for helping setting up the the session for today and the sessions for the next few weeks coming up. And all of you for attending. Appreciate your being here and listening and asking questions. And we hope you'll be here next week with Mason Earls and Concord Earl. So thank you, everyone.
Thank you.